Hi friends, today we will be talking an important, interesting concept called as Central Dogma. Central Dogma of molecular biology. So this is a concept which is there in the chapter molecular basis of inheritance. So we have previously talked about the biology of lac operon. So you can kindly view those video in case if you have not done it till now. Okay. So coming to central dogma, what exactly the central dogma is? So the central dogma seems to be very easy when we look at that particular flow chart in the book, but how do we relate that flow chart conceptually is the thing here. Okay. So let's assume something called as protein. We all know what is a protein, and protein is made up of, it's a combination of 20 amino acids. Protein is a combination of 20 amino acids. We have 20 amino acids in total, and these amino acids are arranged in a particular length, right? Say for example A1, it goes till AN, okay, and in a particular sequence, right? So the sequence and length determines the identity or nature of a particular protein, and we have got n number of protein in our living cell, and every protein has got a particular length in a particular sequence. So this is something regarding the protein. Hope this is very clear. Okay. Now the question is, where does the protein synthesis happens? It happens in an organelle. Okay, it happens in an organelle. So let's assume that this is a cell. Okay, this is a nucleus, and this part will be the, okay, will be referred as cytoplasm. Okay, cytoplasm, and here you have an organelle called as ribosome. So let me try to give a different color. Okay, you have an organelle called as ribosome, and this ribosome is actually the site of protein synthesis. Okay, it is actually the site of protein synthesis, right? Okay, fine. Now let's assume that we try to we need a particular protein. Okay, I'll keep the protein name as P. Okay, this particular protein is required, and where will the protein be produced? Obviously in the ribosome. So we know that particular idea. Now we are expecting this ribosome to make that particular protein. Now the ribosome is going to say that it is ready to make a protein, and in cytoplasm you have the mix of all 20 amino acids, which we call it as an amino acid mix. So there is also that. Now the ribosome is ready to make a protein, but it needs the information. What sort of information? What is the length of this particular protein, and what is the sequence of amino acids? So these two are the information required for the ribosome to proceed with the protein synthesis. And friends, where is this information available? It is generally available inside the nucleus, okay, where we have something called as DNA. Okay, so DNA has got the information regarding the length and the amino acid sequence for that particular protein. That's the reason why we call DNA as a genetic material, right? Now this ribosome should get the information from the DNA. So what do we do? So let's take DNA. Okay, here you have protein, okay, which is prepared in synthesized in ribosome. So DNA to protein. Now this is there inside the nucleus, and you see this ribosome protein synthesis to happen in cytoplasm. And if you look at this previous slide, the nucleus is surrounded by something called as nuclear membrane. Okay, nuclear membrane. Hope things are clear. Now the concept is this: the ribosome has to get the information from the DNA, for which either DNA has to come out of the nucleus or ribosome has to move inside the nucleus. Both of the cases is not possible, because there is something called as nuclear membrane which will not allow either the DNA to come out or the ribosome to get in. So what the nature, how beautifully does manages things here is, the DNA, okay, sends the information for that particular protein through an RNA. So there is something called as an RNA, okay, RNA that is produced, okay, from this particular DNA, and this RNA can associate with ribosome and give the information to ribosome regarding the length and sequence of amino acid with which a ribosome will be able to make a protein. You get this point? Since, wait a minute, okay, now DNA to RNA, okay, RNA to protein. So this is the sequence here, right? Now this DNA to RNA process, we generally call it as transcription. Okay, we call it as transcription, which happens inside the nucleus, because DNA is there inside the nucleus. So transcription also happens inside the nucleus, right? And this RNA, since it acts as a messenger for DNA in conveying the information to ribosome, this is also called as an mRNA, where M meaning messenger RNA. Now this RNA can get out of the nucleus, which we call it as an RNA export, and gets associated with the ribosome, gives the information and makes the particular protein. Okay, the information, again I repeat, it is the length of the amino acid, okay, amino acid chain or a peptide chain, and what is the sequence of amino acids. So these two are the information that is to be given by the RNA to the ribosome in making the particular protein. Okay, now so transcription, and this process is called as translation. Okay, translation. So why this should be referred as a translation or protein synthesis? So protein synthesis is understood because there is a protein actually getting produced here. But why is this called as a translation? What I guess is the mRNA has got a nucleotide. Okay, the protein has got an amino acid. So there is a change in language from nucleotide to amino acid. That is the sequence of series of nucleotide to series of amino acid. Since there is a change in language, maybe as a wild guess should have been had the name translation. So anyone, if you know the information verified, kindly tell it in the comment. Okay, so you have a transcription, you have a translation, okay, and which is also otherwise referred as a protein synthesis. So this is a dogma, central dogma of molecular biology. So information is conveyed from DNA to protein through mRNA. Okay, where you have a process called as transcription and translation, and the DNA is also made to replicate. Okay, and you call this as a DNA replication. You call this as a DNA replication. So how many checkpoints you have here? Let's say you have a DNA replication, and you have a transcription, and you have a translation. Okay, so these three combined to form in this particular order. Okay, and we call it as a molecular central dogma of molecular biology. I repeat, the information flows from DNA to RNA, then to protein. Okay, hope things are clear. And one point to be added before we wind up the session, that in cases like HIV, which are basically a retrovirus. Okay, so there is something called as reverse transcriptase. Okay, there is something called as reverse transcriptase, where RNA is being converted to DNA with the help of the enzyme reverse transcriptase, and the process is called as reverse transcription. Why? Because it is something ulta to the transcription process that we see here. So this is what is called as a reverse transcription. Okay, hope things are clear. Okay, so this is the crisp explanation of central dogma of molecular biology. Okay, if you have any queries, please drop it in the comment section. Okay, let's see in another interesting topic in this particular chapter. Okay, thank you.